Hello everyone and welcome to today's webinar, Simplify Complex Genetic Diseases by Quantifying Structural and Copy Number Variants. My name is Mark Lynch and I'm of Genomic Vision and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Genomic Vision. To learn more, visit genomicvision.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd now like to welcome our speaker for today, Mario Avarello, who's a product manager here at Genomic Vision. Mario, you may begin your presentation.
Thank you, Mark, for the nice presentation, and welcome to you all to the second session of this Genomic Vision summer webinar program. My name is Mario Avarello, and I'm product manager here at Genomic Vision. Today, I'm gonna walk you into the molecular combing for precise detection and quantification of genomic rearrangement at single DNA molecule resolution. I will introduce to you the application of our characteristic genomic Morse codes to level specific DNA regions that are depicted by our FiberVision and analyzed automatically by our FiberStudio software. The use of GMC, such as a molecular marker for understanding of complex genomic rearrangements, such as expansion, shortening, and other complex modification. In addition, we can use GMC to actually understand the genomic variability, such as mosaicism, and it's easy to quantify to the power of our technology at single DNA molecule resolution. I just want you to remember that this is the second session of our summer webinar program. So in the past, you actually learn what are the normal procedure generically of our molecular combing and what are the different steps. So you have some insights about our GMC, about our cancer biology methods and for genetic and rare diseases, and also something for the gene editing. Today, we are going to focus in these to the cases of the use of molecular combing and our GMC for understanding the genomic rearrangement in specific cases, so in specific genetic diseases. And the next summer webinar, in the next session, we'll actually looking forward to have you and walk you through the cancer biology and how to understand the mechanism of cancer and normal cells, while on July 1st is going to be the introduction for the gene editing and how to use molecular combing to qualify and quantify gene editing events. But let's start for this session. Well, we actually will go through the use of molecular combing, GMC, and the entire platform to understand some of the diseases. The first one is FSHD. It's a rare disease and our GMC enable the detection of repeat contractions to facilitate the diagnosis of physicists for patients, while in the second case, we actually use the GMC to see the repeat expansions and quantify these expansions in specific patients suffering of a disease called FAM3. Before starting, I would like to give you an overview about our genomic Morse codes. So the genomic Morse code is actually a molecular marker to hybridize, probe, and visualize specific sequences of the DNA. And then physically, we can actually see what are the genomic rearrangements or the genomic variability that the patient is suffering of. So in this case, you can see that there are different sequences. So we are able to see repeats. We are able to distinguish distances between two different probes or to measure the length. And all of this is possible because we create an exclusive pattern of color. In the first example, you see that there is the sequence of interest, that it's in gray, with the specific sites of the supposed deletion, it's in yellow. So in this case, we actually design probes that are blue, red, and green. And then when there is a deletion, we actually see that the red is getting shorter. In the gene 2, in this case, there is an amplification. So this is the case of the copy number variation. And now the red tract is actually bigger. And in case of inversion, you can see the flip between the colors. Indeed, in the wild type, it's blue, long blue, green and red and short blue. And in the inversion, you can see that it's long blue, red and green this time, and then again the short blue. So these are just few examples of the use of our genomic Morse codes to investigate what are the genomic rearrangements in a patient or in a specific disease. So, but then we can actually see more complex and connected with genomic variability such as mosaicism. This method is actually bring genomic vision to have a strength, be very powerful into the markets, because we actually can see the direct fibers. So we have a direct visualization of the DNA itself or of a specific locus. We can do the direct detection of the locus itself without any technical bias, because we take the DNA, we extract it, we count, so we stretch on a specific cover slip, and then without using any enzymes, we basically probe that specific sequence. That can be any kind of sequence, even GC-rich regions or repetitive regions or large variants that are more than 1 megabase. So this is what the strength of the use of single DNA molecules and our GMC together. But let's start specifically at the summary of the example, and we will focus on the first one, that is indeed the FSHD, and how GMC is able to detect the repeat contraction that leads to the disease onset. So FSHD is actually the fasciculo-scapulohumeral dystrophy. It's a kind of muscular dystrophy. And this is due to the contraction on a specific sequence. There is the chromosome 4q35. There are actually two types of disease called indeed FSHD1 and FSHD2. The two diseases are actually onsets is due to different rearrangements. The FSHD1 is due to a contraction of the repeat expansion, while the FSHD2 is due to mutation in epigenetics genes. So with genomic vision, we actually focus on the FSHD1. So then on the repeat contractions on a specific sequence that is called the 4Z4, in order to understand how many units of this D4Z4 are actually contracted in the disease in the patient. So this D4Z4 is actually present also in the chromosome 4, specifically at the 10q26, and both either in the chromosome 4 or the chromosome 10 in the haplotype A and B. But for the disease, the onset of the disease, the shortening is happening only on the chromosome 4q35, so on the chromosome 4q haplotype A. the use of genomic vision technology with the GMC in a single DNA molecule resolution, it's very powerful because brings some strength compared to the classical method that is indeed Southern blot. So for us, FSHD test is actually one-shot experiment. So in one cover slip, the kind of information that can bring to helping the physician for a diagnosis is actually huge. And this will not require any enzymatic manipulation. So we just, as I mentioned before, comb the DNA and then probe the sequence. So there is no need to use restriction enzymes to amplify the signal for the D4Z4 and then use to classify whether it's in the 4Q, 10Q, whether it's A or B. This will be a unique solution. And indeed, by probing, we can see the D4Z4 directly. We can see in the same experiment whether it's chromosome 4 or chromosome 10, or whether it's the haplotype A or B. We can also see the other rearrangements like the P13E11, and then this will bring to identification of further or more complex modifications such as mosaicisms, complex rearrangements, and even subtelomeric region rearrangement. Some of these are very complicated to identify or nearly impossible with Southern blots. And the last is that we have developed a FiberStudio software indeed to standardize the analysis. If we look at the workflow that is needed to perform our FSHD test, these are the material that we need. So in the first, we have our extraction kit that is called FiberPrep. Is an exclusive kit that uses the extraction via plug preparation in order to preserve the integrity of the DNA. And the fibers, after combing, can go up to 1 megabase. Then the DNA is actually stretched or combed on our specific cover slips that are chemically treated in order that only the DNA ends will bind the surface of the cover slip. And our specific machine called FiberComb is actually the one that's going to make the strength to comb the DNA fiber on the cover slip. Then we use our FSHD probes to hybridize the FSHD signals for the chromosome 4 and then D4Z4. And our automated scanner called FiberVision actually depicts the entire cover slip by taking 25 by 25 pictures and stitching together in order to digitalize the cover slip from a physical one to a digital one. And later, our FiberStudio software will detect and make the semi-automated analysis to identify and quantify the D4Z4 shortening and the complex rearrangements that are happening on it. So looking carefully of how these GMC are designed, we can actually see here that indeed we designed probes for the chromosome 4 and the chromosome 10. So we actually decided to take upstream the D4Z4, and indeed we created the pattern of color that in this case is red, green, magenta, blue for the chromosome 4, and then we hybridized the green tract for the D4Z4, and at the end there is a small tract for the haplotype A when it's red and B when it's blue, and at the very end we have the beginning of the telomere in red. While for the chromosome 10 we use a different code at the very beginning. Indeed we have blue, magenta, blue. This is actually the sign for the chromosome 10. And then again, we have the D4Z4 in green, haplotype A and B in red and blue, and the telomere signal in red. And then is how it looked like in the real picture. So in this case, our scientist has done a study using more than 1,000 patients that were dubious patients from analysis previously done by Southern blots. In this case, we rerun the analysis again by molecular combing and the FSHD probes from GMC. So here, as you can see, this is a real picture of combing experiment, and we have that red, green, magenta, blue is the chromosome 4, and blue, magenta, blue is the chromosome 10. So you can already see the 2D4, D4, and what is actually the length that we're speaking about. And at the end, you see the red dots to distinguish the haplotype A, while below you have the blue dots for the haplotype B, and the red tract at the end that actually will tell us that this is an intact signal. So by using this methodology, actually, our scientists were able to classify 61% of those, out of those 1,000 patients, as non-4Q contracted patients, while 32% were having the contraction. So this is already a good refine of the previous diagnosis done by Southern blots, just using our GMC. In addition, as I mentioned before, it is possible to distinguish for more complex rearrangements, such as a proximal deletion, where you actually see that the blue probe is missing, or a cis duplication, where there is actually a dash between the green and red. And in general, we also looked at more complex rearrangements, such as mosaicism, and in all of these patients, they actually are about the 8%. So in addition to understand precisely and refine the analysis done by Southern blots, by using our molecular combing, we can actually see for the first time and make all these studies to understand what is the correlation of the T4-Z4 contraction with the subtelomeric region. Indeed, we have been measuring the chromosome 4, chromosome 10, and the QA, the distance from the T4-Z4 to the beginning of the telomere. And what we saw roughly is like that there is no much difference in the length between these two subtelomeric regions. But then we look carefully at the subtelomeric regions in the patients having a different length of the T4-Z4, and what we actually see that there is an increased distance between the subtelomeric regions in T4-Z4 repeat contracted patients, so compared to healthy individuals, of course. So this is actually the power again of our GMC that will bring all full insights of what might happen, and will help the physicians to have a better overview to make a better diagnosis for our patients. And indeed, all of these application has been used from the physicians to make the diagnosis of a patient between FSHD1 and probably FSHD2 patients. Indeed, we have, for example, these 4 patients. In the first 2, we actually saw that there is a T4-Z4 contraction, and specifically the first one has 5 units T4-Z4, and in addition, there is a deletion of the P13E11. While the second one has the deletion of the P13E11 region together with 3 units of the T4-Z4. So clearly, these 2 patients were diagnosed as FSHD1 patients. While if we look at the other 2 patients, these again are coming from a dubious Southern blots analysis. We actually found that those patients had 12 repetitive units of the T4-Z4, and there was a deletion in one of them of the P13. So these 2 patients has been classified as not FSHD1, and most probably those belong to FSHD2. So in this case, again, the molecular combing and GMC analysis is a great technology to refine and give a precise analysis and help physicians to classify better patients for the FSHD1 or the FSHD2. And with this, we are at the end of this first part of this session, where I actually introduce you the molecular combing technology and the use of single DNA molecule, and what is the strength of this molecule with GMC for FSHD1 diagnosis in a unique and robust solution. Indeed, I show you that above 1,000 patients that have been analyzed, 32% are actually true FSHD1 patients, while by Southern blot this was still a dubious result. And among of these, 8% also has more complex genomic variability, such as mosaicism, application. And then molecular combing has brought also insights that are not possible to quantify with other technologies, such as the subtelomeric region. And regarding this, we actually saw that between the 4q and 10q, the subtelomeric regions are highly prone to recombine and evolve independently. So this actually opened up to further discussion that would need more research and have scientists and physicians to make a better diagnosis for the FSHD1 patient. In the second part of the webinar, I will actually introduce you to the opposite case where we have our repeat expansions between two introns. In this case, we again use patient samples from a specific disease that is called FAME3. FAME3 is actually familial adult myoclonic epilepsy, the type 3. Indeed, from this disease, there are different kinds of subtypes from 1 to 6, according what is the specific biomarker that is actually bringing to the onset of the disease due to some modification. In this case, for FAME3 is actually 9.3 megabase long, and this is occurring on the chromosome 5p. And specifically is within a gene that's called MARC6 between two exons. We have exon 1 and exon 2, and then we have these intronic parts where, and in red you can see the actual site where the repeat insertion can happen. Indeed, from different studies, what has been looked by the scientists is the identification of the causes from FAME3 onset. So the scientists have been using many of the sequencing technologies, but all were unable to give an outcome. So at this case, they decide to use the RT-PCR. So, and this because indeed you can see that in a normal case, there are the two exons, exon 1 and exon 2, and there is already a repeated expansion between the two that goes from 9 to 20. But in the pathological condition, this expansion is actually even extended more. So, and by the nature of this extension, due to the fact that it's repeated sequences, the sequencing technology was not giving a precise outcome. By RT-PCR, indeed, the scientists have seen that repeat expansion is composed by TTTA or TTTCA into the MARC6 region. So subsequently, once the scientist has known what is causing the onset of the disease, they decided to characterize the disease, and they focus on the use of long-range sequencing, where they could actually see within different patients what are the repeat expansions between unhealthy and unhealthy patients. But again, there was some limitation on it due to the fact that these are repeated sequences. And indeed, you can see from the map where in yellow, within different patients are the intronic sequences that are normally present. And in red, there is the expansions, but that cannot be measured precisely because there is some gap of the sequencing between all these expansions. So in this case, the sequencing could not give a precise overview of the onset of the disease. So at that point, we decided to focus with combing and help physician in make the diagnosis for the patient. And again, we used the strength of single molecule fibers and our GMC to design probes for the two introns, for the two exons in blue and in green, and the intron in yellow. And when there is the repeat expansion, we actually have the expansion in red. So here you can see clearly from the example where it's a wild type and a pathological condition in a real combing. And this has been analyzed in the different patients where they actually could see what is the onset of the disease on these patients, the risk to have the disease on these patients. So again, the FIM3 analysis of single DNA molecules with molecular combing show that the expansions can range from 3.3 kb up to 14 kb. And this is the strength of our GMC and the use of molecular combing together. In addition to have a precise measurements of the repeat expansions that are causing the disease, again, we could look at complex genomic variability that are happening within the patients and leads to have somatic mosaicism. So in this case, you can see in real pictures what are the kind of different genomic variability that we had within the patients. For example, having the repeat expansions at the ends of the exon 2, or where is a combination within the exons. And then we can precisely measure and quantify these rearrangements within the same cover slip between the different patients. So this brought us to actually have that the micro rearrangements can represent the 10% of the allele on a cover slip. And this is the strength of our resolution. So having with bimolecular combing the precise measurement and also a precise quantification of complex rearrangements that can happen in a specific disease or in a specific case. And by this, we are at the end of the second session. So I will just give you the summary where we spoke about molecular combing technology and how can be used in a complementary methods with sequencing. So the single DNA molecule analysis can be used in clinical research indeed to compute without any bias the results that are generated by sequencing or other technologies. And this actually led to have a precise measurement of the number of extension in a specific case that was FIM3 and lead the physician to understand the onset of the disease. And we could actually identify also what are the complex rearrangements that are happening into these patients, intra and inter samples. And again, molecular combing gives a full detail of a patient suffering of a disease and the possibility to have mosaicism that is not that easy to actually see with other methods. By this, we are at the end of this session of the GB Summer Webinar Program. I would like to remind you that on June 24th is going to be the next session that will focus on cancer biology and what are actually the kinetics that bring specifically to the cancer development rather than normal diseases. And by this, I would like to thank you all for your attention, and I will be happy to take questions. Thank you so much.
Thank you, Mario, for your informative presentation. We'll now start the live Q&A portion of the webinar. If you have a question you would like us to answer, please type it now in the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. So with that, Mario, we'll get started with the first question that is coming in. So the first question relates to FSHD, and it is, what is the genome requirement per cover slip for FSHD?
Hi, Mark, and hi all the presenters to this webinar. So just to answer the question, so we actually seen that on a cover slip, we have something about 100 equivalent per genome. So we always start with about 5 ml of blood, that this will allow us to have this quantity. So by using 5 ml of blood, we'll have per cover slip 100 equivalent per genome, and at the end we actually detecting and making analysis for 70 intact signals for the 4QA and the 10QA, and these are already enough to have statistical significant results.
Great, thanks Mario. The next question also relates to the FSHD clinical assay. Can the FSHD clinical IVD assay be used with blood, cells, or saliva?
Yeah, thank you for the question. Really interesting. Yeah, it can be used with blood, cells. At the moment, we don't use saliva. We usually ask to collect fresh samples coming either from the blood or from the tissue from the patient.
Okay. Perfect, thank you. Another question that's came in, other than detecting mutations, can this method be used to view certain alleles in chromosomes to detect certain genetic diseases, such as color blindness or hemophilia?
That's a really good question indeed, because using our characteristic GMC, we actually probed the DNA. So at this point, we actually have a single DNA molecule that are linear. So we normally design probes for the sequence of interest that can be a native sequence or sequence that brings the disease or sequence that has some mutation. But theoretically, we just probe DNA, so this brings us to detect any kind of sequence on the genome.
Okay, perfect. Another question that's came in, how do you avoid the expansions and staining of the non-coding regions, i.e. introns?
We actually avoid the expansions because we don't use any enzyme. So this is just pure count DNA, it's pure DNA that is stretched on a cover slip. And when it's fixed, then we go for hybridization. So there is no modification by using enzyme. So, and in this way, we can actually detect intronic sequences without having any modification of it.
I see. Okay. Another question here for FAME3, can real-time PCR be used for the same type of detection, and how does combing compare to real-time PCR?
They can be used side by side, in our opinion, in order to enrich the analysis in using together. At the time, the paper has been done using a different kind of sequencing or different even kind of assays to identify the intronic sequence first, and then to actually understand how many repeats. So what is the length? And as I mentioned before, the very successful one was using the RP-PCR and then the combing. These were the two techniques that our scientist, our collaborator, has used actually to have a clear result of what are the modifications of the intronic sequences and precisely measure the expansions and whether there are micro rearrangements.
Okay, perfect, thank you. Another few questions coming in, Mario. One relates to the blood being the input for some of the assays with molecular combing. Is the application of molecular combing incompatible with any type of stabilizer or any anticoagulant of the blood tubes that the blood is collected in that you know of?
Well, that's very interesting question. And so far, actually, we haven't had any problem or any effect or side effect with stabilizer, because actually there is a unique way to process the blood from the very beginning. So, and this unique way is actually to start from the blood and immediately, as soon as possible, go to the generation of plaque. And then the DNA actually in the plaque, it's pretty stable and can be kept for even longer than a year. So just following this process, basically, we don't need to, it basically makes the technique compatible with all the other stabilizer or the other method to process blood.
Okay, thanks. So we have one last question that's here in the question field. Some of the images have some background fluorescence on them, and the questioner is asking, would that interfere with the result?
Yeah, that's true. That's true. And the uses of probes actually, and our specific cover slips that are actually, would like to remind us that have they are coated chemically in order to allow the DNA ends to bind it, actually retain sometimes probes. So this might increase a bit the background, but actually we have developed the Fiber Studio software that allow us to have a precise detection of the signal. And then we usually design the GMC in order to have any avoidance, to avoid any problem or any bias coming from the background. So each probe, each segment that you see has a specific length, and we know. So, and putting together the detection of the software and the length of each segment, each color, we actually are sure that there is no bias introduced by the background itself.
Understood. And we have time for one more question that's just coming in. Since this method is very new and really unique, how compatible is this for everyday experimental use?
Yeah, that's a good question. This is a really new method, as you mentioned, and we are improving our technology, and we are always changing our software in order that will become more automated and more precise for the detection. So this brings us to have a really high throughputs, and that can be used in everyday in lab. And also our machines, as mentioned before, for example, the FiberVision, the automated scanner, can actually host up to 160 samples, and it takes about 160 minutes for each cover slip. So at the end, it's actually a quite good method that can be used for every day in lab.
Thanks, Mario. So we don't have any other questions at this time in the box. Mario, do you have any final comments for our audience today?
Thank you, Mark. Well, no, I don't have any other comments. I will just thank you all for their attention, and I would like to bring again what is the strength of the use of molecular combing to identify specific sequences on the DNA that can be mutated, can bring to some diseases. And with this, I'd like to thank you all for your attention, and I will look forward to meet you all at the next session on July 24th, where we'll actually focus more on cancer biology and the tools that we use to understand the mechanistic role of cancer, of the replication in cancer, and how it can be used for the drug screening.
Excellent. Thank you again, Mario, for your time today and your important work here that you've presented. We'd also like to thank LabRoots for facilitating today's educational webcast. Before we go, I'd also like to thank the audience for joining us today and for their interesting questions. It was very interactive. For any questions that we did not have time to get to today, or for any that are submitted during the on-demand period of the webinar, these will be addressed via Mario through the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage that you share that email with your colleagues who may have missed today's live event, and we look forward to seeing you at part 3 of our webinar series on June 24th, Improved Quantitative Understanding of Oncological Replication Kinetics Using Molecular Combing. Until then, thank you and goodbye.